Hi everyone, my name is Jenna, and in this video I will be talking about the Pfeiffer lab and their work on cell junctions and cytoskeleton. First, what are cell junctions? Cell junctions are protein structures that act as a bridge connecting two cells together. This way, they make communication and exchange of material possible between cells. There are different types of junctions present in cells. Our main focus in this video is adherence junctions. Specific purposes of adherence junctions are to provide mechanical strength to the cell, prevent cells from separating, and allow them to send signals to each other. A cytoskeleton is a cell organelle that is composed of protein fibers that maintain the cell's shape. You can think of it as a dress cage that maintains the dress's shape. Its purpose is to provide strength like cell junctions, allow for cell movement, and move organelles within the cell by acting like train tracks. When connecting two cells together, junctions attach to the cell cytoskeleton to form a bridge between the cells. This bridge is important for tissue development by preventing cells from separating during this process. A biology lab here at UNC interested in this particular topic is the Pfeiffer lab. It is associated with the Lineberger Comprehensive Cancer Center and is led by Dr. Mark Pfeiffer. As mentioned, the relevant areas of interest of the lab include cell adhesion, which discusses how cells connect and interact together, as well as how cytoskeletal behavior is regulated. In this lab, Dr. Pfeiffer conducted research trying to identify what proteins actually make up the cell junction and cytoskeleton bridge. Dr. Pfeiffer achieved this using a technique called proximity proteomics. This method involves using a protein as a tag, which in this experiment was BRA. The target protein that was tagged was afidin, which was found in cell junctions and is essential for maintaining tissue structure. Once the afidin proteins in the model cells were tagged, all proteins within a vicinity of 10 to 15 nanometers from the tagged proteins were collected and identified. This list of identified proteins was then compared to lists created in previous studies with other types of model cells. From this experiment, the lab was able to identify 144 different proteins. This result suggests that cell junctions are extremely complex and include a wide variety of proteins. When compared to results from other experiments, the list of identified proteins in this experiment had both overlapping and non-overlapping proteins, as shown in this Venn diagram. The big takeaway from this study was that the cell junction and cytoskeleton bridge have a complex network of diverse proteins. Another conclusion was that cell junction composition varies between different cell types while they share some common proteins. Moving forward, scientists can use this information to discover the role of each identified protein when forming bridges between two cells. Ultimately, understanding this process during tissue development helps scientists understand the formation of cancerous tissues and use proximity proteomics to identify early signs of cancer for effective diagnosis. Overall, Dr. Pfeiffer's work at this lab makes valuable contributions towards a better understanding of cell-to-cell interactions in cancer research. And that is the end of this video. Thank you for watching.